everyone. Myself, Cytometer, I'm a PhD scholar at School of Biotechnology, Kidd University. We'll be telling about our very own flow cytometer. So I'll be detailing the basic principle, how flow cytometer works, what is flow cytometer, what we can do in this flow cytometer. So first of all, there are going to instruments. So flow cytometer basically works with three principles. One is fluidics, second one optics, third one is electronics. So first, coming to fluidics, so we have this setup named as fluidic setup. You can have a look here. This is fax flow. We call it as fax sheath also. It's a sheath fluid because, as the name suggests, flow cytometer. Cyto means cell. Cell will move and meter will count. So how cells will move? By the help of this fax sheath. So we call it as fax sheath. The machine which we are using is from a company named BD, and the model number is BD Fax Canto 2. The company from US, and it's a very famous company and one of the pioneer in flow cytometry. So in this fluidics system we have fax flow, one more solution named it as fax clean in the back, and one more we call it as shutdown solution fax rinse, and one more we have one tank here that is known as waste tank. So there are three liquids involved for successful running of this instrument. So what are these liquids actually? So as I said earlier, we have fax flow for the simple running of our cells, for the movement of cells. In this flow cytometer we are using this sheath fluid. For example, suppose we have a cell pellet. We will take the cell pellet, we will dissolve our cells by the help of this sheath fluid. Then by using fax tube we will run the sample in this flow cytometer. So that is the use of fax flow. Next we have fax clean. The second one, fax clean. Exactly as the name suggests, it will clean the flow cytometer after a single run. So why it is necessary? Suppose in a hospital, there are 1,000 number of samples are coming. Some are cancer positive cells, some are cancer negative cells. So after each run we need to clean the flow through. Flow through means through which this sample will pass in this flow cytometer. So for cleaning after each run we use fax clean. So the basic composition, mainly, although it's a patented product, but the basic composition is maybe the 1x PBS with some antibiotics and detergents. So we have one more solution named it as fax rinse or shutdown solution. So what is that? As this is a flow cytometer, so many tubings are present inside this machine. So tubings are like always filled with a liquid substance. Why? Suppose after my run I am switching it off. So once I will switch it off, my tubes will be vacant. So there is a chance, suppose I have used a bacterial sample or any nanoparticle or any cell, so there is a chance of blockage of those tubings. So if there will be vacant space, those tubes will be blocked. So just to avoid that condition, we are using one solution, fax rinse. After the successful run of our experiment or after our all samples run, we will, by switching off the machine, the shutdown solution will go inside and it will fill the total tubing inside the flow cytometer. So this is about fluidic system. I'll say one more aspect of this fluidic system, how exactly these fluids are going inside this machine. So you can have a look here. There are many tubes, those are connecting this machine. So you can see the color code now. This is orange colored mark. So here it has clearly written also waste. So all the waste samples through these tubes are coming to this tank. And now you mark this color and you observe the color code here. These tubings are also colored with orange color. So this orange color indicates these are all waste pipes. Those are collecting the liquids from the instrument and passing these liquids to this waste tank. So now this is about waste. Fat seal, as I said, this is the main liquid which will help us in running our sample. So fat seal, you see the color code in the packet. This is blue colored mark. So see another color code in the instrument. So this is also blue color. So this seed fluid is going to this instrument by the help of this nozzle. So this is seed nozzle, we call it as, and label it as blue color. So fat stream and fat streams are going by these tubings in the down. So next, as this is a flow cytometer, there is a constant pressure in the machine. So we are maintaining the pressure through this tube by giving air pressure. So here you can also see labeled it as air in. So through this white tubing, air will go inside. That will maintain the pressure while we will run the sample, while we will count the sample. After fluidics, next comes optics. As the name suggests, this machine will count different number of cells with different color mark. Suppose we will stain our markers with specific color, for example red, blue, green. This instrument can mark those colors. So how it is counting those colors? We have 3 lasers specifically for this instrument. So we have 3 lasers. If I remove it out, we can have a look in this person, although it is tightly fitted, but we have 3 lasers in this position. So these 3 lasers will, now this laser will come, and in this position we have one prism. So in this prism 3 lasers will bombard and one monochromator wavelength light will pass through. So this light will pass through this channel, and while giving this different wavelengths as different color we are using, we have different wavelengths for each specific color. Suppose for example red, that is 495 wavelength nanometer, 495 nanometer. For FITC we have 488 nanometer. So how our instrument will recognize those wavelength marks? So behind of this laser we have 3 setups known as filters. We have 3 different types of filters. One is short pass, long pass and band pass. What is the name short pass? Short pass, as the name suggests, suppose this wavelength, suppose this is the filter which we are using, has a cutoff range of 500 nanometer. So for short pass, the wavelength which is lesser than 500 nanometer will only pass through. As the name suggests, short pass, and the cutoff is 500, only the wavelengths which are less than 500 will pass through, rest will go back, means that will be reflected out. So that reflections will be captured by another filter. We have a series of filters, so that wavelength will be captured by another filter. So maybe that is a long pass. What is the long pass? As the name suggests, suppose we have a cutoff range for that filter is 500 nanometer. So the wavelengths more than 500 nanometers will only pass through that medium. Then next is our band pass. Band pass means Band pass means suppose we have a 500 nanometer filter, then suppose we have a cutoff range plus minus 50, then in that case 500 plus 50 wavelengths will pass through and 500 minus 50 wavelengths will pass through. So how these wavelengths, why these reflections, from where these reflections are coming? So actually when we run the sample, we are using different cells. Actually I'll say you just now why we need these samples, why we need this instrument. Why this instrument is specifically analyze different types of, suppose, cancer cells present in a mixture of cells. We can differentiate those cells by staining specific cancer cell markers present in that cell. So for an example, in a hospital we have 10 patients. So we'll take the blood sample from 10 patients. We'll analyze those 10 blood samples. So we'll stain our cancer positive markers with a color. So that color, that percentage we can calculate here. How many patients are having cancer positive cells and how many patients are of cancer negative cells. So actually when we are running these cells, single cell has a single like structure, shape, size. So when this single cell will pass, this laser will bombard the cells. When this laser will bombard the cells, based on the size and shape, there will be reflections. So some will pass through this, some will go, some will reflect out. So the lasers which are like bombarding the object and moving forward, we call it as forward scatter. The laser will bombard here and move in a 60 degree or a 55 degree or a 70 degree angle. We call them as side scatter. So generally forward scatter as FSC, forward scatter, and side scatter SSC. So from this forward scatter and side scatter, this instrument will identify each population cell. Suppose for an example, you are taking a blood sample. So in a blood sample there are like mixture of cells. Neutrophils are there, basophils are there, eosinophils are there, and all those basophils are of similar type. Neutrophils are of similar type. So these neutrophils will give a similar pattern of reflection. Basophils will give similar pattern of reflection, and based on those reflections, this cell population will be counted by using this software. So before going to the software, as I am telling this optics part, so there is like one more very important aspect with this instrument is this nozzle. So what is this nozzle? This is a like solid bar here. This nozzle, this can take one cell per, like at a time only one cell can pass through. So this is a very sensitive instrument. So we need to make our sample will diluted so that at a single run we get single cells will pass. So there will be perfect counting of those cells. Because we can control this flow also. As I said before, we are maintaining the pressure by the help of this air in. So how this air in is playing the role? Suppose I'll give one example. This is the fax tube. This is the fax tube which we will run for our sample analysis. So this fax tube will get fit into this nozzle. So once we will fit it to this nozzle, then we will remove the staff. So this will hold our fax tube. Suppose we have a mixture of cells here. So when we have taken this out, now this air pressure will be generated. When we switch on the machine, this air pressure will be generated. Once this air pressure will be generated, cells will move up. Now you imagine we are giving equal pressure from the other side. Now cells will move through this nozzle. So this air pressure we can control also. So there is a like option, we can give medium, high and low. So based on our cell density, we can control the flow of these cells. So next, after the cells will pass through this nozzle. Through this nozzle, as I said before, these wavelengths are coming, like lasers are coming through this prism, and they will bombard the cells. So once they will bombard the cells, then scattering patterns will be there. Forward scatter, side scatter. Based on those forward scatter, side scatter, populations will be counted. So for a flow cytometric analysis, there has to be 10,000 number of cells. That's the minimum required number of cells for analysis. So you are going for a publication, you are like minimum statistically like significant data we can calculate from minimum 10,000 cells. We can go beyond that, but not less than 10,000. So we'll run the sample. So this is with optics, forward scatter, side scatter, lasers will come, cells are passing, they will bombard, scattering will be there. We can capture those scattering patterns by using a series of filters, band pass, short pass, long pass. Then this optical signal is getting transferred to an electrical signal. So fluidics, optics and electronics. So next we'll go to the electronics part. So how these optical signals are getting converted into electronic part. So we'll analyze that. So for this instrument, we have a setup here. We can control this instrument by using a software named as Fax Diva. So now I'll switch on the software. So in a flow cytometric analysis, there are many things coming. So we have taken a big window because there will be an analytics like worksheet, one cytometer, one browser, one acquisition dashboard. Everything will be. I'll come to that. So before, suppose this is Fax Diva software. I'll double click it. When I'm double clicking it, this window will come. So this window starts BD Fax Diva software. As I said before, it has 3 lasers. 3 lasers can read 8 different colors. So 3 lasers, by moving through these different wavelengths, short pass, band pass, long pass, that will give us 8 different colors. So next we'll go to the view option. So once we click the view option, there is an option named as browser. We'll click the browser. When we click the browser, this window will come. So in this browser window, you can name your experiments, or you can name by your name also all your experimental details. So for example, I'll open a folder. Suppose this person X has a series of experiments. So these experiments can be recorded after each run in this browser setup. So next will be a Cytometer. So this is a Cytometer window. Next, view, worksheet. Worksheet will come to this window. All are in inactive stage now. I'll say how to make it activate. So next is view, acquisition dashboard. Okay. Suppose I'll make a folder now. I'll do an experiment now. In my folder, there is an option now. This floppy symbol beside my folder. I'll click this floppy symbol. That I can rename now. So this is giving experiment number. So I can rename it with my experiment. Suppose my experiment is apoptosis. I can name it as apoptosis. Next, see, another mark is activated now. When we click another mark, that stands for specimen. What exactly treatment, what exactly you are doing. So in your experimental folder, you can make few subfolders also. Suppose this specimen I can name it with apoptotic drugs. Means I have used few apoptotic drugs and now labeling them in this folder. Next, you can see one more option here, tube option. When I am clicking this tube, two options get activated in this down. So I'll delete one option. So this is my first option. I can name it as control. So this is the setup with browser. So browser, we can name our experiment, what exactly we are doing. We can name it as suppose apoptosis assay. So in apoptosis assay also, what you are doing, another option is coming in the name of specimen. We can label it as with apoptotic drugs. That means suppose I have used different types of apoptotic drugs and I am checking the activity. In that case, I will use that apoptotic drugs folder. Next is my tube, means my experimental conditions now. So first one is control. I have labeled it. Now next I will go to second window, that is cytometer. So in this cytometer you can see four sub columns now. So if I keep my cursor here, this corresponds to fast flow, first column, and it is giving green. Green signal means my fast flow tank is fine. That means sufficient amount of fast flow is present in my fast flow tank. Next is second one is waste. So my waste tank, I have discarded the previous waste. My waste tank is now vacant. Third option is shutdown solution. We have shutdown solution option. As I said, 3 fluidics. Last one is fax clean. That means the cleaning solution. So everything is fine. If there will be any problem, this will be in red color mark. That will indicate us, yes, there is a problem in this flow system. So next we go to parameters. So once we click this parameter window, there will be many sub columns now. So first one is FSC. You can have a look here, FSC. As I said before, FSC stands for forward scatter. The next one is SSC, stands for side scatter. Based on this forward scatter and side scatter, we will analyze our sample. As I said, it can read 8 colors, so you can have a look on those colors. So this is split C, fluorescein isocyanide, the green color, P, phycoerythrin, red color. So we have another 5 colors also, named it as Percipi, Cy5, P, Cy7, APC, Cy7, Pacific blue, and Amsat. So what are those colors? Suppose in our experiment, we have taken only one color, means we have used only one antibody. For an example, we are staining our cell by using that antibody, and that antibody is conjugated with a secondary antibody that is coated with a blue color or red color mark. For example, red color. So antibody with red color will bind to our specific cancer cell. Then this cell with the marker with the red color antibody will give us red fluorescence. So we can capture those red fluorescence. So while suppose we are analyzing only one color, then we should delete the rest of these colors. So we'll delete answer by clicking this delete option, delete. Pacific blue we can delete, APC size 7. Similarly we can delete all the colors, but we'll keep only FSC, SSC and P. Next, in these parameters is done, we have set our parameter, we'll read the color that is red in color, and our FSC is ready and SSC is ready. So next will be our third window. This window is known as acquisition dashboard. So when we click this acquisition dashboard, so there are many marks here. So as we have set this control, so this is saying your control sample. 0 event per second. This is called threshold rate. 0 event per second means this instrument calculate each cell as each event. So how many cells are moving per second, this will indicate us when we run a sample. So 0 event per second. As the system is idle, we are not running any sample now, so this is showing 0 event per second. Then stopping get events means after like how many cells will should stop. As I said, for a statistical data, we need to at least have 10,000 number of cells. So we can set this number by using this software. This is events to record. So this is 10,000 event now. I can increase the event by clicking here. This is 10,000, 20,000, 30,000, 50,000, 1 lakh, 10 lakhs. Similarly, we can calculate the number of cells, but the minimum number cells is 10,000. So I'll keep my cells count to 10,000. So why we are calling it a stopping gate means after 10,000 cells, system will automatically stop the cell flow. Means only 10,000 cells will move. Those 10,000 will analyze. Rest all we are not going to take. So this is 10,000 number of cells. So fourth window, this is global worksheet. This will get activated when we are going for the analysis. So before going to run, I'll tell you, as I said before, we can control the flow, we can control the movement of cells. As I said, we can make it low, we can make it medium, we can make it high also. So by clicking in this acquisition dashboard, there is an option name here. We can click. So there will be 3 options: low, medium and high. So once this low, medium and high will be there, based on our cell density, cell population, we can control this. This is the controlling of those air pressures so that cells will move freely. So for a normal run, suppose I am keeping this in medium mode. Okay. Next we will run a sample. So before running a sample, this is about the instrument, how it moves, how it works, what are the requirements, how exactly this instrument is working, fluidics, optics, electronics. But before that, why we need this machine? Why? What we will analyze here? What is our main principle? Why we are requiring this machine? So I would say you can do wonders with this instrument. So what exactly we are doing in this instrument? Suppose for an apoptotic dead cell, you can differentiate between a normal cell and an apoptotic cell. Suppose you want to check any specific expression inside the cell, you can stain those proteins with specific color, then you can count those cells. So this is like very sensitive technique. You can count those cells, you can tell the population, like population of cells. Next, suppose for an example, you take the blood sample. Blood sample contains different types of cells. So many, like the blood sample we generally consider it as a heterogeneous mixture of cells. In normal cell culture, we are using homogeneous mixture of cells. Homogeneous mixture of cells means only single type populations we are using. The cells which we are using are of single type. Suppose macrophage, we are culturing homogeneously only macrophages. So when we will take those homogeneous cells, that can be like counted by having three graphs. One is of dot plot, second one is by contour, third one is by histogram. So dot plot, I click this dot plot option, I can draw one graph here. As this instrument analyze two-dimensionally, so XY graph will be there. X-axis codes for forward scatter and Y-axis codes for side scatter. So this is forward scatter and side scatter. Next, suppose we are having a stain sample, red color stain sample. We will make another dot plot in the down. One more dot plot. We can name it as PE. We can clicking in the x-axis, we can set this P range. As I said before, we have taken only one color, PE, the red color, phycoerythrin. So in x-axis coding also there are three options coming, FSC, SSC and P. As we have taken SSC in y-axis, we are taking PE here. So this is for the basic understanding of the software. So first we need to take a graph of FSC and SSC that codes for the population of cells, single cells. Next we are giving this with PE population. Only PE positive cells we will count. So this count can be done by three different ways. One can be of dot plot, one can be of contour, one can be by using histogram. So for an example, I'll delete this dot plot, I'll make one histogram here. So now we will run a sample. So this is the fax tube. I have taken 2 ml of sample in this fax tube. Now I'll use this tube and I'll use this machine to read those samples. So I'll remove the sac and fix the fax tube, then I'll again remove the sac. So once we are fixing this tube, next we'll control this machine by the help of this fax diva software. So now in this, because we are now acquiring the data, so in this acquisition dashboard we will click acquire data. So once we are clicking it, now you can have a look. Initially it was 0 events per second. Now you see the number is changing. 16, 32 events per second, 40 events per second. That means this many number of cells are now passing per second. 48 events per second. See, in this graph also, there are few black dots are coming. As I said before, this is dot plot. We'll get dot for our single cell, and this is the histogram. Histographic plot we get for our sample. So now you have a mark here. This FSC has linear scale, 50, 100, 150, 200. Similarly, SSC has also linear scale. Now cells are moving. Population we are getting, but somehow we are not getting a single population. Population is coming in a line. So what we will do, we will change the unit of this x-axis. So we can change the unit in this cytometer option. So we click cytometer. There is an option named log scale. So we click log in case of FSC. Now you observe the FSC line became in log scale. Next in SSC also I'll click. So SSC became in log scale. Now you have a look. I am getting a very nice dense population. As I said before, we are using homogeneous mixture of cells. So we are getting a single population of cells. And there are some breakage during sample preparation. So we are getting few populations out of these main populations. So this can be minimized by making a gate around this population. So before going to the gating system, I would say one more thing. As I have used one homogeneous mixture of cells, so we are getting a single population. So if we are using a heterogeneous mixture of cells, so in that case we will get different types of population, different populations in this single XY plane. So that indicates our sample is fine, we are getting a single population, and we will now proceed with our further analysis. So how we will get those samples? Because these are some unfragmented cells, like fragmented cells, and this population is of my interest. So in this worksheet, I'll go to polygon gate. You can have a look here. This is the polygon gate. I can click this polygon gate. Now I can draw a gate here. So once I am drawing a gate here, so this will indicate us a population named it as P1. So now you see the color is changing. The population which we have taken now became red. That means the population of our interest is inside this boundary. So this population is named as P1. So you can have a look. This is giving a nomenclature as P1, means population 1. So we will analyze population 1 only. Next, here also we are getting some extra. This is the same representative image but with this P line. This was with FSC line, forward scatter line. Now we are given the fluorescence wavelength and fluorescence mark here. We will analyze our sample with PE film. So as I said, our population is P1. So I'll click a right click here. I will give a like show population. Sorry. I'll give a right click. I'll take show population. That will be P1 only. So you can see the difference now. Here it was the initial population from where we have selected our specific population. Yes, the dense one, the homogeneous cells. Okay, I have taken this P1 population. Next, in the second window, I have taken those P1 populations only, and I have removed those extra black areas. Next, this is also same representation of dot plot. We are getting in histogram. Here also I will say show population P1. So these two are similar data, but representation is different. This is dot plot, this is histogram. Next, this population is coming here. So what to do next? So I can adjust my population also. I can adjust my population by the help of PMT voltage. PMT stands for photomultiplier tubes. So where this voltage is coming, we will go to cytometer next. So in cytometer, you can have a look. Here voltage mark is coming. This is the voltage for FSC, 300 voltage is for SSC, P for 500. So in this voltage, I can like increase the voltage, I can decrease the voltage. So based on this increase and decrease, I can move these populations in this XY plane. So why that is necessary? Because somehow we are getting population here. Suppose for an example, you are getting population here. So this is your control population. Next will be your treated. So there will be more Pi positive cells, more P positive cells. So then your population will move out of your graph. So initially your population was here. After treatment populations came here. That means your population is moving. In that case, you cannot count those cells. So for that, we need to bring my population to this origin. So how I bring my population to origin? Because if I bring my population to origin, even if there will be shift in y-axis, in x-axis, I can analyze. So I can move this population by the help of this PMT voltage. So nothing I am changing, only I am tuning the position. So for like in a normal television, we can control the volume. So we can somehow increase the volume, we can decrease the volume, but we are not changing any other parameter of the television. Similarly, here we will only change the position of cells. So by the help of PMT voltage, now you see I am moving this. So this is P500, I can move it little down. Now this is 346. So I will click it. Next I will restart it. When I restart it, now you see initially population was here, now it is going towards your y-axis. Now you can say no, I want my population in this origin, very close to your 0 position. Okay, we can bring this population down also. So you see the mark now, SSC. So we can change the volume of SSC. So when we are making it down, we'll make it down slowly. So once we are, next our job is to restart it. So when we are restarting it, so now you see your population is coming down. But somehow, so next, as in our analysis we have selected a population. So this is our population of interest P1, and these single dots are single cells. So we have taken this P1 population, and in our PE window we have taken only P1 population. So next we will record the data. Now this is accurate. So next step is to record the data. So as I said, we are taking 10,000 events, means 10,000 cells will count. So I will click the record data now. After clicking, now this count will increase. Now you see 81, 108, 189, 243. So this count also I can control by using the flow movement. Now I can make it little high. Now you see population is moving faster. Now I can make it little low also. Now you see populations are moving very slow in that nozzle. So for a better analysis, always it's recommended to keep a medium flow rate so that all cells will pass through. And this is 36 event per second, means 36 cells are moving per second, and we have already counted 2,500 cells. So next we will count for 10,000 number of cells. So you see the mark here. So after 10,000 event, system will automatically switch it off. So once it will switch it off, we can remove the fax tube. We can keep our next tube for analysis. So as I said that we are using different set of samples. So after each run, we need to clean the nozzle. So I will show you after this run, when I am removing the tube, this liquid will come, this fax clean will clean the nozzle. So you can hear the sound, although it's very thin, but you can have a mark on the sound. But after removing the tube, you can see how this nozzle is getting clean. So now already we have counted up to 5,000 cells. So this is now 5,076. So now this instrument has already taken like 5,000 cells for analysis. Now 5,000 new cells are coming. So from that fax tube. So I'll increase speed little high. Like now this is moving very faster. So you can have a look here, 7,000. This time is indicating how much time machine has taken to count these many number of cells. So it is saying 2 minutes 19 seconds. Yes, we are approaching towards 10,000. It's done. Now 10,000 events. Now you can have a look in this fax tube. The sap has come down. So we will take the sap towards left. We will remove our fax tube. So when we release the sap, you can have a look now. Instrument has taken the sap. Now you can hear. It means instrument has cleaned this nozzle. It has already cleaned. So this instrument is now ready for our second set of samples. So this is how we analyze our sample. So this is with one sample. I am giving you a demo. Suppose you have a series of samples. Next job is to make a gate here. So this is our control sample. Next will be our treated sample. I will take another treated sample here. I will fix it here. This is my second sample with treatment. I will make the column now. Next tube, I will make it treated. Next, similarly as before, I will acquire the data. See, the count has started. Now I will record. Once I will record, this number will increase. Now you can see it's counting very fast. 300, 400. So this also will count up to 10,000. So this is our treated sample. Initially it was controlled. Now treated. After this treatment, now this is stained with PE. We will make a gate. So we will keep a gate. From that gate, we will calculate how many PE positive cells are there. I will tell you how we can make that gate in this software. So after 10,000, after this run, we will make a gate in this graph. So after the second run of our treated sample, we will make a gate here. So from how we will make this gate? So there is an option here, interval gate. So we can take this interval gate and we can click it here. So once we are clicking it, so there will be like two gate now. One is P2, one more gate is P3. That means the P2 population is of, suppose we are comparing this treated sample with our untreated sample, means the unstained sample where PI was not there, P was not there. So this will indicate how many cells have taken that dye and have moved forward. So PI positive cells will move forward in this direction, in this x-axis towards right. So when I am making this gate, now I can get the percentage value. How I will get the percentage value? I will click on this graph, right click, then next will be show population hierarchy. When I am clicking the show population hierarchy, now you can see P2 population 44%, P3 population 52%. Means this P3 population, blue color, when I am comparing with unstained, have moved forward. Means 52% of cells are positively stained with red color and 44% of cells are not stained. Means those are only cells, normal cells, not stained with the right. Suppose you are using a cancer cell. These 52% are of cancer positive cells, 44% are not. So based on that, we can get the quantitative data for a given sample. Yes, 52% and 42%. This is how a flow cytometric data is different from a microscopic data. Microscopic data gives us a qualitative analysis, qualitative data, but in flow cytometer we can get quantitative data in terms of percentage. So this is how we calculate our sample based on our different color code. So this is the basic principle how this flow cytometer works, how we are making this game. So this is with a single analysis. So many things you can do. You can check the reactive oxygen species formation. You can check pore formation. You can check the expression of different proteins, cytoplasmic proteins. You can also check the different expression of surface markers. So we can stain those proteins, we can stain those things with our specific marker and we can analyze. For example, cell cycle also we can do. Our cell cycle has 4 phases, G1, S, G2N and G0 phase. We can calculate how many percentage of DNA are in G1 phase, G2 phase, S phase. So we will make 4 different grades. So 4 different grades will give us 4 different stages of DNA. So cell cycle analysis we can do. We can check Ross chromosome. We can check apoptosis, necrosis. So apoptosis, necrosis also we can check. We have specific antibodies for an axin 5 FITC which will stain our phosphatidylserine. It will bind to our phosphatidylserine, that will give us apoptotic cells, and our PI that will enter inside the cell and bind to our nucleus, that will give our necrotic cells. So based on these PI positive cells, FITC positive cells, we can also differentiate between early apoptotic cells, late apoptotic cells and necrotic cells. So this is how we can use this instrument in our day-to-day research for the betterment of our analysis and the better data which we can publish in a highly reputed journal. So this is how the system works, from fluidics through optics to electronics. So fluidics, optics, electronics, and we are controlling this instrument by the use of this FACS DIVA software. We are analyzing so many things by giving gauge. We can control the air pressure, we can control the flow rate, we can control the PMT voltage, we can control the movement of cells, we can count those cells. So many things can be done by the help of this flow cytometer. So we have come to the last part. I would say thanks, a big thanks from Kids School of Biotechnology. So next will come with a new set of instruments, with a new set of data analysis, with a new set of approach towards research. Thank you very much.